Hello, welcome back to this course, Cell and Tissue Organization. Under Unit 2, we will continue our discussion on cell structure and function, and then end the unit with the discussion on the movement of substances in and out of the cell. So under Unit 2, Session 1, we will be looking at two other important organelles, that is the cell vacuole and Golgi bodies. By the end of this session, it is expected that you should be able to differentiate between the types of vacuoles and their functions, and also describe Golgi bodies, and then also show how the Sistani maturation model of Golgi bodies differs from the ordinary model, and also lastly to note down two functions performed by Golgi bodies. So starting with vacuoles. Vacuoles are fluid-filled sacs bounded by a single membrane, and they are present in all cells but comes in different sizes, and they perform different functions. So the animal cell vacuoles are relatively small as compared to the vacuoles that are found in plant cells. An example of vacuoles are the food vacuoles, contractile vacuoles, and cell sap vacuoles or central vacuoles. So the food vacuole is formed when amoeba and many other protists feed by engulfing food particles and other smaller organisms. So the particles that is engulfed found in the cytoplasm becomes a food vacuole, and it becomes digested by enzymes released by lysosomes. Now, contractile vacuoles helps in the regulation of water content of the cell, that is osmoregulation. An example is in Paramecium, which is a protist, and these contractile vacuoles helps in pumping excess water out in order to maintain a suitable concentration of ions and molecules. In plants and fungi, the vacuoles carry out hydrolysis in addition to other roles. So plant cells have large central vacuole containing fluid called cell sap, and they come in varying concentration and mixture of salts, proteins, gases, and waste materials. The central vacuole is developed by the collection or coming together of smaller vacuoles derived from the endoplasmic reticulum and Golgi apparatus. So the membrane of the vacuole is referred to as tonoplast, and it is selectively permeable, and so they ensure that the cell sap differs in composition from that of the cytosol. Now let's look at the functions of vacuoles. So vacuoles serve as reservoir for organic compounds and inorganic compounds. And some vacuoles also serve as disposal sites for metabolic substances or byproducts that will be harmful to cells when they accumulate in the cytosol. And some vacuoles contain pigments that give cells color to the cells. So some vacuoles also contain poisonous or unpalatable compounds that protect plants against their predators or animals. So in plant cells, vacuoles contribute to the growth of the cell, and this is because the new cells or new plant cells that are formed, the vacuoles absorb water for the cells to become bigger in size, and so the pressure in cell subvacuoles also enables the plant to support itself. And some vacuoles also control the water content of the cell by pumping excess water out of the cell, and that is osmoregulation. Now let's have a look at Golgi bodies or Golgi apparatus. Golgi bodies were discovered by an Italian physician called Camillo Golgi, and these Golgi bodies are found in eukaryotic cells, but they cannot be found in prokaryotic cells. So Golgi bodies or Golgi apparatus consist of flattened membranous sacs, that is cisternae, containing secretory granules. And then the membrane of each cisterna in a stack separates its internal space from the cytosol. Figure 7 gives a structure of Golgi bodies, and these Golgi bodies are surrounded by a cluster of vesicles, and these vesicles function to transfer materials between parts of the Golgi and other structures. The Golgi stack has two poles, and these are the cis and the trans poles. And then the cis pole is located near the endoplasmic reticulum, and they act as the receiving department, whereas the trans phase is responsible for shipping of materials from the Golgi body to other structures. And one thing that we have to know about these two poles, that's the cis and trans phase, is that they differ in thickness and in molecular composition. The vesicle of the Golgi breaks and joins onto the cis phase, and then the Golgi enzymes or enzymes that are found in a Golgi, they modify the carbohydrate portions of glycoproteins during the transit from the cis phase to the trans phase. The Golgi also removes some sugar monomers and substitutes, and others also produce large variety of carbohydrates. So the trans phase of the Golgi apparatus gives rise to vesicles, and these vesicles cleave off and travel to other sites. So non-protein products also depart and are transported via these transport vesicles from the trans phase of the Golgi apparatus, and they fuse with the plasma membrane. So this model of Golgi is referred to as cisternal maturation model, and this cisternal maturation model postulates that secretory vesicles that travel in the cisternal compartment slowly mature from the cis phase of the Golgi to the trans phase of the Golgi. And then Golgi is known to produce or is involved in the production of cell membranes, cell walls, and then the cuticles of insects. Some also contain digestive enzymes, example being the lysosomes. And others also are involved in the secretion and packaging of complex carbohydrates and proteins. So under Unit 2, Section 1, we looked at two organelles in the cytoplasm of cells, being the vacuole and Golgi bodies. And we studied that vacuoles are single membrane-bound sacs that are filled with fluid, whereas the Golgi apparatus are flattened membranous sacs containing secretory granules. And then the Golgi apparatus are also involved in secretion and packaging of carbohydrates and proteins. And then the different types of vacuoles was also studied, and we got to know the function of each of the vacuoles. And then the main function of the cell sap vacuole was also known to be osmoregulation, whereby excess water is pumped out of the cell in order to regulate the water level or the quantity of water in the cell.